At this point, you have collected your osmosis data, and now you're ready to write your lab report. So let's talk about what that entails a little bit. No matter which lab section you're in, if you're in my lecture, you should be doing about the same thing. So first things first. This report is over the osmosis experiment with the potatoes. You don't include the stuff you looked at with a microscope. So Elodea is not on there. Tetrahymena is not on there. If you missed lab, let's say you're quarantined, right? You still have the lab data because the class collected lab data. If you're in my class, at least my sections, you have access to that Google Sheet. If you have a TA that collected data on the board, you can get it from them by contacting them. You're still responsible for the lab report. Now, if you go to the lab website, you can go to the live pre-lab video. So if you missed lab this week and you don't understand what happened, you need to watch the live pre-lab. So in normal weeks, right, we have pre-labs, and you're all familiar with pre-labs. Pre-labs are these things where you watch a video over osmosis, for example, right? You've probably done that to get ready for class. But the live pre-lab is when I film myself giving the lab. The first lab of the week, I record myself teaching it. That way, if you miss, you can watch it and catch up. So you have the data and you have access to the Osmosis Live pre-lab, so you can watch that and know what happened in lab, at least as well as I can provide. Okay? Now, let's talk about the report itself. It's going to be about 1 to 1.5 pages long. You should follow the instructions in the appendix of your lab manual. There's a whole appendix on how to write a lab report and a rubric for how it will be graded. And in that how to write a lab report section, there is information on font size, on margin sizes, things like that you should pay attention to. And you also read a Piranha paper, which will give you ideas for how scientific papers are written. You don't say, I did this, typically, and you don't say, you should do this. You don't usually see the words I or you in a scientific paper. They're typically written from the third person. So 25 milliliters of sucrose was added to a test tube is how you would typically write from that perspective. And you want to be concise, clear, etc. You've watched the video by Dr. Runge as part of your lab online lab assignment. So you should have a pretty good idea of what goes into a scientific paper. So let's talk a little bit about the osmosis report itself. You're going to have an introduction. And that introduction is going to include background. So it's really important that you have background, just like a pre-lab would have background. And this experiment was not about potatoes. This experiment was about osmosis. Potatoes are just the model we use to understand osmosis. So what is osmosis? Why is it vital for life? These are the things you might write about. And you're going to have to cite your textbook when you talk about that. And use the format that I show you in the lab manual. How does it work? What does it do? Describe the process. Give background. Pretend that the reader of the paper isn't really familiar with osmosis terribly well, so you need to explain it to them. Why are potatoes a good organism to study? Well, that one is just mostly because they're available in this case, but you can explain why they'd be a good one for this study. What is the research question that you're asking? And what is your hypothesis? So what was it you're trying to solve, and what was your hypothesis in the if and then format? Okay? And that takes care of both of these bullet points if you use the if and then format, essentially. Materials and methods. How was the experiment set up? How was data collected? And make sure you cite the manual. You don't have to write the size of all the beakers or anything like that. It's just a description of what you did, but it's not written as a list. It's written in paragraph form. Potatoes were cut into 5 millimeter thick discs and placed into very concentrations of sucrose. And then you would tell me what the concentrations were, and then you would say procedures were followed as in the lab manual, and you would cite the lab manual. Results. There is a paragraph here where you're going to describe your major findings. Not interpret them, but describe them. and tell about the overall trends. What do you see? By trends, I mean, what was the overall outcome here? What did you see overall? If you collected any qualitative data, you want to talk about that. And you want to reference your figure, in parentheses, figure 1, at the ends of sentences where it's important to do that. You also need to have your figure in there. That's a graph and caption. Conclusion. This is one of the more important parts of the paper, and it may well be the longest part of your paper. It should be, usually anyway. That in your introduction. Discuss the results of your experiment in light of your hypothesis. So, did the results cause you to support or reject your hypothesis? What quantifiable evidence can you present in defense of your position? What results weaken your defense? So talk about the data. How do you interpret it, and how does it strengthen your argument, or how does it weaken your argument? Explain why the data happened then. I don't mean by saying when potatoes were put in sugar they lost mass. That's not an explanation. That's a description. Why did it happen? From the molecular level. Don't just say osmosis happened. Explain what was going on with water molecules and sugar molecules to cause those trends to occur. Do this thoroughly. Do this well. Explain it. Also, what concentration was the unknown? I have that on this guide in the conclusion. I would probably actually move that to the results section. What concentration was the unknown, and what concentration would be isotonic to your potato? Both of those require you to solve the question using the equation of the line from your graph. So a hypotonic solution would have 0 change in mass. And the unknown, you know what change in mass it had. So calculate what the concentrations would be for those. Let's see here. What is the importance of your findings? Relate them to biology as a whole. So relate them to medicine, or to health, to IVs, etc., or diabetes. Relate it to something relating to biology. What future experiments could you do to follow up on this one? So think creatively. If I had next week's lab was a blank slate, and I said, you're just going to take what you learned from osmosis and from that experiment, and you're going to build your own experiment to further your knowledge, what would you do? So write a little paragraph about that. And you're going to have a literature cited section as well at the end. Now, this guide here is not something you should just fill out and then you got an A. This is a guide to help you out, but you need to pay attention to the rubric to make sure you're not leaving anything out. You need to write clearly and concisely. So work on your rough draft. Get that rough draft written. Turn it in by October 2nd. And then visit the Writing Center as soon as you can to get feedback on that rough draft and revise it and edit it and turn in your final draft. By the deadline, I think it's like October 16th or something. So get that turned in, okay? All right, so that is the lab report in a nutshell. And I posted on Blackboard an example lab report as well, just to give you an idea of what we're kind of looking for. So do a good job. Put in time. If you just phone this thing in, you're going to end up with like 22 out of 50 points. You got to do a good job, all right? And you can do it. I'm not trying to scare you. I'm just saying don't phone it in. Do a good job. Edit. You should never turn anything into an instructor that's not been proofread and revised. Make sure that you've read it over at least 5 times before you turn something in, even the rough draft. All right. I'll see you in class.